One of the most basic questions in biology is, what are you made of? Now, living things, including your own body, are made of five main ingredients. The first one is water. It's the most abundant molecule in your body. It plays several important roles in all living things. In fact, life as we know it doesn't even exist without water. But for now, we'll focus on the other four main ingredients, which are the four classes of organic molecules. They're the carbohydrates, the lipids, the proteins, and the nucleic acids. Please note that the term organic means that these molecules are made of carbon as their backbone. In this podcast, we'll summarize the structure and the functions of these four groups of organic molecules. The first class of organic molecules is the carbohydrates. Carbohydrates are sugars like glucose, and they're polymers like starch and cellulose. In terms of structure, they can be recognized as being made of multiples of CH2O, hence the term carbohydrates, which means carbon with water. For example, glucose, the most abundant carbohydrate, has the formula C6H12O6. Glucose is a monosaccharide, which means single sugar. Two monosaccharides can be joined to make a disaccharide. For example, if you take glucose and fructose and attach them together, you get sucrose, or table sugar. Sucrose is a disaccharide. Several monosaccharides can be attached to form a polymer called a polysaccharide. For example, starch or cellulose are both polysaccharides made of glucose. Carbohydrates play three main functions. The first one is energy storage and processing. For example, photosynthesis in plants produces glucose, and in turn we eat this glucose and transform it into a usable form of energy. The second important function of carbohydrates is structural support. For example, cellulose, this polymer of glucose that was mentioned earlier, forms the bulk of wood. And the third main function of carbohydrates is cellular recognition. Carbohydrates on cellular membranes are used by your body to recognize your own cells. This is how the ABO blood groups work, for example. The second class of organic molecules, the lipids, is a group of nonpolar hydrophobic molecules. Hydrophobic means that they don't dissolve in water. They contain several types of molecules, but we'll cover the three most important here: the fats, the phospholipids, and the steroids. The first group of lipids, the fats, are made of three fatty acids, which are long chains made of carbon and hydrogen. They're highlighted in yellow here. So three fatty acids attached to one glycerol molecule. The glycerol is in the gray shaded box here. The properties of fats vary based on the nature of their fatty acids. If all the fatty acids are saturated, that means there's no double bond between the carbon atoms and the fatty acid chains, then the fat is said to be saturated, and it will be solid at room temperature. Think about butter or animal fat, for example. If the fatty acids are unsaturated, as shown here, that means there's at least one double bond between the carbon atoms, then the fat is said to be unsaturated, and it will be liquid at room temperature. We call these fats oils. Think about olive oil or fish oils. Fats and oils are used primarily for energy storage, but also for thermal insulation and mechanical protection of various organs. The second group of lipids, the phospholipids, is structurally similar to fats. They're made of glycerol as the backbone, to which two fatty acids are attached. Instead of attaching a third fatty acid like in the fats, here we find a phosphate group to which a variable hydrophilic group is attached. So a phospholipid is a glycerol molecule, two fatty acids, phosphate group, and variable hydrophilic group. This confers an unusual property to phospholipids. They're both hydrophobic and hydrophilic at the same time. Their tail, made of the fatty acid chains, is hydrophobic, while the region with the phosphate group is hydrophilic. It dissolves in water. Such molecules are said to be amphiphilic or amphipathic. Because of this amphiphilic nature, phospholipids will tend to form bilayers in water. That means two layers of phospholipids forming a sort of sandwich with the hydrophobic tails in the middle. This property is actually essential to determine the structure of biological membranes, as we'll see in another podcast. The third group of lipids that we cover today are the steroids. In terms of structure, they're all made of 4 fused rings. Steroids play a variety of functions. Some will stabilize cellular membranes, for example, cholesterol, which is shown here, while other steroids are important as hormones, for example, estrogen and testosterone. The third class of organic molecules found in all living things is the proteins. Proteins are the most structurally and functionally sophisticated and diverse molecules known. In terms of structure, all proteins are polymers of amino acids. This is what an amino acid looks like. There are actually 20 kinds of amino acids that can be used to make a protein. For some amino acids, they all share the same basic structure. The only thing that changes is what's called the side chain or the R group, and it's highlighted in the white box here. A protein's made of a few up to several thousand amino acids attached in a chain. The chain will then fold to take a variety of shapes. The specific sequence of amino acids making a protein is called its primary structure. This primary structure, the sequence of amino acids, determines the final shape of the protein and therefore the function of the proteins. Proteins are responsible for pretty much everything in living cells. They're responsible for at least most of the work in living cells. So some proteins will provide structural support. Think about the collagen in your skin or the keratin of your hair. Other proteins are involved in movement. Think about the actin and the myosin in your muscles. Some proteins transport substances. Think about hemoglobin, which transports oxygen in your blood. Other proteins carry signals, for example, insulin, which is used to control your blood sugar level. Other proteins are involved in your immune system as antibodies. A specific type of proteins called the enzymes catalyze chemical reactions. Now, these enzymes are responsible for most of the metabolic reactions in living things. They're extremely important. The fourth type of organic molecules found in all living things, the nucleic acids, store and transfer genetic information in cells. In fact, they contain the information to make specific proteins and are involved in the synthesis of these proteins. Nucleic acids are DNA and RNA. DNA, or deoxyribonucleic acid, is the type of molecule that contains your gene. That's the information to make specific proteins. DNA is found in your chromosomes, in the nuclei of your cells. The information contained in DNA can be used to make proteins using a fairly complex machinery. This process actually uses the other type of nucleic acid, RNA or ribonucleic acid. We'll cover the functioning of DNA and RNA in more details in another podcast. In terms of structure, DNA and RNA are both polymers of nucleotides. Now, a nucleotide itself is made of 3 things: a sugar, a phosphate group, and a nitrogenous base. Now, the sugar is called ribose in the case of RNA and deoxyribose in the case of DNA. It's a pentose sugar, which means it contains 5 carbon. Pentose means 5 carbon sugar. The phosphate group is pretty simple, and the other component of a nucleotide is the nitrogenous base. There are 5 types of nitrogenous bases: adenine, or A, thymine, or T, cytosine, or C, guanine, or G, and uracil, or U. Thymine is found only in DNA, and uracil is found only in RNA. A DNA or RNA molecule consists of a chain of nucleotides. In a DNA or RNA molecule, the sugar and the phosphate group are always the same. The only thing that changes along the length of the molecule is the nature of the nitrogenous base. The variations in the nitrogenous bases is what allows DNA and RNA to carry genetic information. The letters corresponding to the nitrogenous bases, A, T, C, G, and U, are the letters of the genetic alphabet. Now, one more thing. RNA is a single strand of nucleotides. DNA is made of two strands facing each other in opposite directions. They're said to be antiparallel, which means side by side but in opposite directions. What holds both strands together is the hydrogen bonds, which are weak interactions that will occur between A and T and between C and G. So, if the nucleotide on one strand of the DNA molecule has A as a nitrogenous base, then the adjacent nitrogenous base on the other strand will be T. A can only pair with T, and C can only pair with G. So A and T are said to be complementary bases. C and G are also complementary bases. Finally, this double-stranded structure of DNA will twist to form a double helix with the sugar and phosphate forming the sort of the backbone and the nitrogenous bases in the middle of this double helix. Now, the structure and functions of nucleic acids is an entire topic on its own. Once again, we'll see in another podcast how the structure of nucleic acids is related to their function. Okay, I hope this podcast was useful. Please check the blog for more podcasts. The address is biopodcasts.blogspot.com. Talk to you soon.